Continued flagella. The flagella of bacteria are powered by a different source than the flagella of eukaryotic cells. The attachment of flagella at one or both ends of the organism is called polar distribution. If the flagella are distributed over the entire surface of the organism, this is paratrichous distribution. The distribution of flagella is one means used to differentiate between types of bacteria. Tactic behavior. Prokaryotes demonstrate specific tactic behavior. They can swim in response to specific environmental stimuli. For example, they can move toward or away from light, phototaxis, in response to oxygen, aerotaxis, or to a magnetic field, magnetotaxis. Chemotaxis refers to the ability to swim to a useful food source or nutrient, or to swim away from a harmful substance. Fimbriae or common pili. Pili are short hair-like extensions on the surface of the cell consisting of protein. They are shorter and stiffer and of smaller diameter than flagella. The sex pili mediate the transfer of DNA between mating bacteria. The more numerous common pili, usually called fimbriae, provide adherence to the surface of the cell. This is a major factor in bacterial virulence, which allows pathogens to attach to body tissues and colonize into groups. It also allows bacteria to resist attack by the body's defensive white blood cells or phagocytes. Chromosomes, DNA. Bacterial cells contain a large, naked, double-stranded, round molecule of DNA, genetic material, floating free within the cytoplasm. The cells may also contain pieces of DNA called plasmids. The total of all DNA is called the genome. Bacterial cells do not undergo meiosis or mitosis as do eukaryotic cells. Replication and segregation into two identical cells is coordinated by the cell membrane. Ribosomes are involved in protein synthesis in a different way than in eukaryotes. They give the granular appearance to cytoplasm. Inclusions are usually materials being reserved for future use by the organism, such as glycogen or fats. As stated, bacteria can be identified by their flagella, how they move, Gram-Stein reaction, and their relationship to oxygen. In addition, bacteria are identified by their shape. Most bacteria fall into one of three categories based on shape. A round or spherical bacterium, a coccus, plural cocci, or cocci, coccus or cocci. Rod-shaped is a bacillus, or bacilli for plural, and spiral-shaped a spirillum, plural spirilla or spirochete. The cells of cocci do not always separate when they reproduce. Sometimes they do form pairs, diplococci, clusters, staphylococci, or chains, streptococci. Likewise, diplobacilli are paired bacilli, and streptobacilli are chains of bacilli. Bacilli may have tapered ends, fusiform bacilli, or may be shaped like long threads, filamentous bacilli. Bacteria may be classified according to their flagella, motility, Gram-Stein result in relationship to oxygen, as well as their shape. Unlike eukaryotes, such as protozoa, bacteria do not reproduce by binary fission. In bacterial cell division, genetic material is duplicated and distributed loosely to daughter cells, a process, genetic recombination, coordinated by the cellular membrane. There are three means by which bacteria exchange genetic material. Conjugation, DNA crosses a sex pilus to the new cell by cell-to-cell contact. Transduction, genes are transferred by a virus. Transformation, DNA is acquired from the environment after being released by another cell. The implication for disease control relates to the fact that bacteria usually develops genes for drug resistance. Resistance transfer factors are RTF on structures called plasmids. The plasmids are often the vehicle for cell-to-cell DNA transfer and are able to pass drug resistance to other duplicate cells, other strains of bacteria, and even to other species during the exchange process. Protective spores are formed by certain bacteria. Examples are Clostridium tetani, which causes tetanus, and Bacillus anthracis, which causes anthrax. When conditions are unfavorable to their survival or growth, spore-forming bacilli develop a protective covering or spore and go into a non-active or dormant phase. The spore is resistant to the environment and can survive extreme conditions of light, drying, boiling, and many chemicals. When more favorable conditions return, the spore germinates and the bacteria cell reactivates. Spore-forming bacteria are the most difficult bacteria to control and destroy. All pathogenic bacteria die at water's boiling point, 100 degrees Celsius or 200 degrees Fahrenheit, except for the spore formers. Many bacteria are able to cause disease. Pathogenicity. To be pathogenic, bacteria must possess the following characteristics: ability to colonize and invade the host, ability to resist or endure the antibacterial defenses of the host, ability to form substances that are toxic to the host. Neisseria are diplococci and cause gonorrhea, upper respiratory infections, and infectious meningitis. The bacillus, Pseudomonas, is responsible for suppurative or pus-forming infections.